Hello, everyone, and welcome to the April 2026 CB seminar. And I'm going to turn things over to Jeff Tomberlin to introduce our guest speaker.
Excellent. Well, first, good morning to everyone and good afternoon, Jeroen. It is my pleasure to introduce someone that I consider not just a colleague, but a friend. And it's exciting to have an opportunity to learn about the research that he's been conducting in his lab overseas. Jeroen graduated as a bioengineer. Major was in cell and gene technology in 2011. He obtained his PhD in 2016 at the Laboratory of Gene Technology on the exploration of phage-host interactions. He is married and a proud father of three children. And in 2017, he moved from the campus in Leuven to KU Leuven campus Kiel and turned his research focus towards microbe insect interactions during insect production within the research group for insect production and processing, which is IPMP. After two successive FWO postdoc fellowships, he applied successfully for a BOF research professor position. And since 2024, he started as an associate professor to build his own research line, which is a micro insect with IPMP. For this research, he also received a startup grant from the Francois Foundation. The overall aim is to map, manage, and mine microbial communities associated with industrial insect production. To date, his research revolves mainly around understanding microbial diversity and dynamics in the gut of industrially produced insects and their impact on insect performance, with the black soldier fly as the model organism. Jeroen, the floor is yours.
Thanks, Jeff, for the nice introduction and for the invitation. I'm very happy to indeed share some of the research I've been doing. So the coming 45 minutes, I want to introduce you to some of the research that has been going on within our team, or the team that I am leading, which is the microsec team within our research group. But first, indeed, some more about our research group. As Jeff already said, we are located in Belgium, in Geel, which is to the north of the country. And we are actually a multidisciplinary group in that we have three PIs that are collaborating very closely together, of which I am one. And our mission is to strengthen the insect value chain as a cornerstone of the circular bioeconomy in Europe. But of course, we have global interactions and research collaborations as well. And on our campus, we are quite uniquely located in the sense that we also have our insect pilot plants, which, if you are interested, you can take a virtual tour after the seminar via this link that is on the screen. So it's actually a very unique building that was uniquely designed to facilitate pilot scale insect production, but also houses an L1 and an L2 biosafety lab, which allows us to combine a lot of microbial research with insect research, which before that was quite difficult. The research within our group has actually three main focus according to the three PIs. So there is one more on chemical safety and also chemical extraction methods. There is the one that focuses on the microbiological safety, and that's the one where I'm mainly involved in, which I will tell you more on later. And then there is the third research focus, which is more on processing and how we can use or process these new matrices in a safe and sustainable manner, and how that impacts their safety and quality and stability. So most of the focus today will be, of course, on this microbiology, because this is where I am more focused on. So this goes from assessing microbiological safety, but also trying to understand the role these microbes play. So why do we focus on this, or what is our focus? So if you look at insect production, you can of course look at your insect, but we believe that it's very important, which is since I started my first postdoc in the lab, is that we actually, when you look closely at the system, of course, we talk about industrial insect production, but we have a number of factors that are very important and are very closely interacting. And at the time that I started my PhD, there were more and more studies coming out about insects and testing them on different substrates and seeing how they behave on these different substrates. But at that time, I felt that there was this third component, the microbiota, which was back then, I think it's much improved by now, not that much discussed, like how do the microbes both inside the insect, but also inside the substrates change or interact with these different components. And it's only by understanding this triangle and also how this triangle interacts with the environment that you can really start predicting performance or improving performance. So we set out to really try to solve these question marks of what are these interactions and why are they important. And this all frames within this holobiont concept, which you hear in many different organisms. Also in human research, it's very important that we are, as an organism, of course, we're not solely ourselves. We have a lot of microbes inhabiting our system, and these microbes can also play important roles. This has already been shown for different insect species that they play key functions. For example, if we think towards nutrition, it can be that microbes can help us process substrates to deliver key components that the insect itself cannot produce, for example, vitamins in mosquitoes, but they can also help in protection, be it against abiotic stresses, chemical stresses, or biotic stresses such as predators. These are different examples, so there are reviews on this from other insect species, and there's no reason to think that the same is not true for industry-produced insects. But we still lack a lot of fundamental knowledge on that. So that's why we think it's very important. This is, for example, highlighted by the example of a study by Andreas Lemme and Patrick Kluber from a few years ago, where they actually wanted to study the amino acid requirements of our black soldier fly larvae using traditional animal nutrition research. So what you typically do is, according to Liebig's law of the minimum, you are trying to see which one is your first limiting amino acids, because if we know that, we can also make sure that the right amounts are ready so you can have maximal performance within your diet. But then you need to know these requirements for each of the amino acids. And what they noticed in their research is actually that when you typically do in this kind of research is you reduce your amino acids to see when something becomes limiting. But what they saw is actually that when they reduced amino acids, they did not necessarily saw an impact on the growth of the black soldier fly larvae. So these reductions somehow seem to be alleviated, and their hypothesis, which they could not show, but they explain in the article, is that microbes which are present in the substrates are able to help and recover these losses, and as such they play an important role in compensating such reductions, which of course complicates also the research in trying to understand what are your requirements. And as such, it is a very simple and elegant example, in my opinion, of why we need to study this microbiota and understand the roles it's playing. And this is only one example. I'm, of course, not the only one. If you look back, since one of the first articles I published in the insect research domain in 2018, there are numerous reviews, very comprehensive ones in there as well, that discuss the microbiome of insects, be it the black soldier fly or more broadly, and they all highlight that there is already some knowledge being gained, and there are studies that indicate important functions of the microbes, but that we are still lacking a lot of information that is needed. So the domain is clearly growing, which is of course good, because this is typically a field where we need a lot of data to really come to a comprehensive understanding of the system. The challenge in the amino acids requirements is only one. I think there are other challenges in insect nutrition, and these are the ones I wanted to highlight a bit in my seminar today, where I do think that this microbial component matters and why we think that the research we are doing is hopefully very relevant for the insect sector. So, of course, the first major hurdle in insect nutrition is, of course, that we have a novel livestock species. So we are not really clear on what are the nutritional requirements. In traditional livestock species, we can rely on decades of nutritional research to sort of map what is actually required. In insects, we do not have that background already. There are some recent studies that try to combine what we know already. For example, within the Bug Book Initiative, there was one on nutritional requirements. And there we do see some general conclusions on, like, okay, these are the same amino acids that are relevant for mammals are also relevant for insects. And we also have these sterols, minerals, B complex vitamins that are essential as well. But often there, already one thing is that who produces these? Do they need to be present in the substrate? Can microbes produce them? These kind of questions, of course, then remain. But trying to get more information on that is challenging, because if you think of traditional livestock nutrition, often the research is quite linear in the fact that you have your feed, which has a certain composition, you know your input, you get your animal and animal performance, and you get your feces, so you can have these very clear mass balances of what is going on. But of course, if we think about our insects, or especially the insects most of us are working on, which are the mealworms, black soldier flies. This kind of linear system is not really the case because our insects, they live in their substrates. So the feces becomes part of the feed becomes part of the feces. There can be microbial interactions going over different feeding cycles. So it's a much more complex system to understand, and you need to know your microbial component because this might change over time your substrate composition, which can be also important for your performance. So knowing your microbiome is important as well. At the same time, there's a bit of a conundrum there in the sense that you want to have a very defined diet because then that's how traditional animal livestock is. If you know your exact requirements, you can define your diet as such and you can have maximal performance from your diet. But if we look at insects and how we envision insects as a sort of a key factor in how we can increase our sustainability, well, there we see that if we look at, for example, this LCA analysis, which was done by Sergei Smetana in 2019 already, well, we see that we can have a much bigger impact on this sustainability if we actually not use these very high quality diets, but rather use these non-utilized side streams. They can even cut the impact, according to this calculation, in half. So that means that a highly formulated diet might not be your best way to go, and we then need maybe less formulated diets. But then, of course, how can we optimize these? What roles can microbes play? Again, microbiome information is very important in this. And a final point I wanted to highlight, where we are also more and more looking at, is that we see these different business models more and more becoming apparent, especially in Europe. So we had our centralized insect production system where we have this one very big facility close to maybe a large food source. For example, if we think of InnovaFeed, which at one central location produces a large amount of microbes. And on the other hand, you have your decentralized model where you have your breeding facility, which distributes neonates to different breeding sites, which then can use locally available side streams and produce your insects there, and then maybe bring them back to a central facility, but it's far more decentralized. But in both cases, if you look at them, there are actually interactions between this nutritional research, because, for example, in the centralized model, you might want to work with a nursing phase diet, which is very high quality and can ensure a good start vitality for your neonates. But then in your fattening phase, you might want to switch to a more highly abundant organic side stream to increase your profit and sustainability. This will, of course, not only have an impact on your insects because they have to be able to work on both diets, but of course, it might also be the case that there are different microbiomes present or microbiomes required to aid in that digestion. And is it possible to shift? What is the impact of such shifts? That's also research that one of my PhD students is tackling. And the same can be said for the decentralized model, where you have a central location, which may be a very high quality diet because you want to give good neonates to your customers. But of course, if they have very different diets with very different microbial requirements, there might be these kind of interactions there. So although it is crucial, I think, that we can work with our black soldier fly with these different diets, because that's the way that this insect will be important for a more sustainable food system, it does require some optimization, and it can translate into some friction between what you want from a traditional livestock production system and from a more waste conversion system. So these kind of challenges, they require fundamental research on this microbiome. And in my group, I've tried to align this in 3 research lines, where we have one where we look at the fundamental insights in our microbial dynamics and diversity. So really trying to get these fundamentals understood, because we need those to translate them into actually manipulating our microbiome, which means we want to develop new microbiome engineering tools that we can use. So as already said by Jeff, mainly for now in the black soldier fly model. And at the same time, these fundamentals will, of course, also lead to insights into functions delivered by our microbes, which might be relevant for completely different fields as well, if we think of antimicrobials or novel enzymes for fiber degradation and so forth. So I will not have time to show you all of these things we've been doing. I do wanted to highlight mainly the mapping and the manipulating side. So what we have been doing on mapping the microbiome. First of all, when I entered as a postdoc, I wanted to try and understand how the microbial community is structured in black soldier fly, meaning who's there. I think that was also the first reflex of the entire field. So we also contribute to that in the sense that we built our own culture collection where we looked at over 170 isolates that are abundantly present in our black soldier fly on different substrates. And we saw that actually 5 genera represent over 90% of this library. And if we did then the other way around, where we did a meta-analysis, by looking at all studies that were available at that time, so that were about 11, and we looked at these results from this 16S rRNA gene sequencing. Also there we saw that there were roughly 5 genera present in over 70% of our samples, meaning that they were also quite conserved, in this case, over different research fields, different research studies, and so forth. And if we then compare them, we see some names like Klebsiella, Enterococcus, Providencia popping up in both our analysis. And if you now look at the more broad reviews that are, or more recent reviews that came out, these names still hold true. So there seem to be some genera that are quite often associated with our black soldier fly. There was a few years ago this discussion about the core microbiome, or is it completely flexible? So that was this first attempt we did in that field. Once we have an idea of how the microbiome community is structured, of course, you also want to see how how robust this is and what is the impact of different parameters on it. So we've done also a lot of studies where we mapped how external factors influence the structure of this community. So over time, we looked at time, location, diet change, the virome which is present, and these QR codes lead you to some of the articles we've published on these topics. So I will not go into detail in most of them. I do want to highlight one of them, which is recently been accepted for publication. So it's not out yet, but it's more deals with the genotype. This actually stems from a collaboration we have with our colleagues at Thomas More, who are also located at our campus. And the question we asked there is that if you have different genotypes and you have them at one location for several generations, what happens to these bacterial communities in these same environments? Do they completely merge? Do they stay uniquely different? Because this might impact if you start selection, or you want to select the best strain for a specific diet. Of course, if there's a microbial component, you cannot really select only on genetics, so it's important to know. So we got from our colleagues of Thomas More these 10 different strains, which were bred for at least 6 generations in our insect pilot plant. And we did a 16S rRNA gene sequencing analysis. And just here you can see one depiction of the results. So what we try to do here is show which OTUs, which maps to a specific microbe being present, was actually most abundant in our samples. So abundance you can see by the size of the bubble. So the bigger the bubble, for example, this set OT2, the more abundant this microbe was in our community. And then you have the color, the darker blue, the more shared it was over these 10 different strains. So you can immediately see that there's a lot of blue in our figure, meaning that over these different generations being reared at the same facility, a lot of the microbiome does indeed converge, and we see the same microbes being present in these different lines. But at the same time, there is still some red present, and red means that they are quite unique to this specific genetic strain or only shared by a few of them. So even after these, let's say, 6 to 10 generations, which is relatively a long time, in this similar environment, we still have these small differences, which does hint at some impact of genetics on host microbe interactions and which microbes can establish or can persist in these kind of insects. So that's very interesting, we think, for further fundamental research. At the moment, we are still exploring this further in the sense that we have now set up a trial with these 10 strains, but putting them on 3 different diets, and then seeing if there is a different adaptation in the microbiome to the different diets for each genotype. So the sampling has been done, and currently we are doing DNA extractions, and I hope that by end of summer we can have some more details on that going forward. So trying to wrap up a lot of research over the past years, if we come back to the question, do we have this core microbiome for our black soldier fly, or are they rather like this blank canvas that can be colonized by microbes from the environment? Well, the data we think at the moment hints that there is some retention of specific bacterial genera, meaning that some genera are more likely to colonize our black soldier fly gut, but there is a very high impact of the environment at the same time as well. And this is most likely the case because it also is something we see in other insects that inhabit these unstable habitats, for example, Drosophila, where you have this limited obligatory association, because then you allow flexibility and you create the opportunity for choosing the most beneficial microbial partners from the environment, which we believe is also something that might be at play within our black soldier fly. However, it still does not mean that these core genera we see are then not very suited for these roles which our bacteria, our insects are looking for. So that's why we do believe that it's quite crucial to better understand what these roles are. And that brings me to our currently biggest ongoing line within this mapping of the microbiome, and that's trying to go one step further. So a lot of this has been these large sequencing-based mapping attempts, but now we want to really go into studying the role and the fate of the microbiota and specific key bacteria, for which we have been developing some different methods, technologies, setups, let's say. One which I will not go into detail today is trying to get collaborations going with the group for livestock technology to have automated monitoring available in our group for both larvae and adults, because we believe on the long term, if we start modulating our microbiome, it would be nice that we have high throughput, reliable methods to monitor what is the exact impact on performance rather than only having an end weight analysis or so forth. So that's something that, thanks to the collaboration with Professor Arnout on our campus, is something that I hope to continue on. Another aspect is setting up methods to better track gut transit dynamics. So this is research that originated from our exploration of chemical safety. The chemical safety line, we looked at microplastics, how these microplastics were taken up in the gut. But from that research, we saw that these labeled plastics are very interesting to see what is the size that can be taken up, how long they reside in different gut environments. So also in collaboration with Moritz Gold from ETH, we've done a few research articles on this front, looking at these plastics, seeing how gut transits react to diet composition, which is something that's also part of a PhD student's still ongoing research. And also now we want to take that one step further. By using labeled microbes, we could do the same, but then actually looking at the microbes, how long do they reside, where do they reside in our system? But more on that later in this seminar. A third important aspect is our way of how we understand what a specific microbe is doing. Because if we look at our microbial community, it's still quite complex. So it's often very difficult to understand what one specific microbe is doing. And one of my postdocs, Nate, has been working hard these past years on setting our axenic and gnotobiotic model. So in literature, you already find some articles that have claimed such a model and in their own lab, but we've noticed that it was quite difficult to translate that to our research facility, also to see whether you can get full development from your larvae, that they actually still grow, that they develop into adults. But by now we can state that we do have a functioning model. So I wanted to elaborate a bit on this more, because we think that these models are very crucial tools to really go into the role of the microbiota and the role of key bacteria. And I want to give an example on that. So when we talk about the role of microbes, we do find claims in different articles that say, like, for example, Providencia is linked to protein degradation. But when you go into detail, okay, how this has been proven, often this is based on bioinformatic analysis. So meaning that you use, for example, tools like PyCrust, which links genomic data, meaning you have with 16S RNA gene more frequently isolated your Providencia in these conditions. And from the genomic data, we know that there are more metabolic pathways associated with protein degradation in this kind of a genome. So that must mean that this insect or this bacteria plays a role in protein degradation. So it's still very, circumstantial, let's say. So there's a high risk of overinterpretation, especially what I notice if one study cites the previous one and then cites the previous one. So there is still more work to be done on really trying to understand what, for example, the Klebsiella, the Enterococcus providentia is doing there. And we believe that as long as you are working with this very complex, undefined microbiome, it's quite challenging to really see what is this function. You could enrich one specific bacteria, but it might still be hard to understand what is especially then changing and if it's really linked to that one bacteria. We hope that by working with this sterilized eggs and rearing these sterilized eggs on sterile substrates, that we can have our axenic model, meaning that there is no microbiome, which already allows you to see what you are missing because you have no microbiome left anymore. And then you can go one step further by introducing your selected microbe and seeing how then certain functions might come back online. But this is, like I said, this is something we are now currently just taking our first steps in. We did, however, already work with our axenic model, and indeed it shows that we can use this to show the importance of our microbiome. So what we see is that the development does happen if we work with our sterilized chicken feed. So we do see that we can get up to adult flies. Currently, we are confirming that they can still produce viable eggs, but we can get full cycle. We do see, however, that there is a reduction. So these are quite new research results from NATE, where we have seen that indeed you see that there is growth. They might even get up to a higher size or mass than the standard rearings, but it does take them roughly 10 days longer to reach this same mass as your standard rearing ones. A key side note here is that we see that whether you have made really axenic ones or you use sterile feed, that the difference there is not that significant. So it does seem to be that the microbes in the substrates are very important to take into account. Using this model further, we can also prove that these microbial functions are clearly context dependent. And we've done so by testing two different diets. So we have used chicken feed and wheat bran, which if you look at the composition are actually quite comparable in fat content and protein content. But what we noticed was that when we transfer our eggs to a sterile nursery diet, which was in both cases chicken feed, and at the same time we prepare both sterile and non-sterile chicken feed and wheat bran, and we then transfer our neonates to this experimental substrates. What we then actually saw is, if you look for the chicken feed, after 21 days, that whether you worked on this fresh chicken feed or the sterile chicken feed, there was not really an impact on growth. However, in the wheat bran, it's a completely different story, and there we saw a clear impact of this sterilization on growth performance of our larvae, meaning that in this diet, they are missing certain functions. And what we then try to find out is whether this might be microbiome or microbial functions. So to test that, we went one step further, which you can see here. So here you see that two additional conditions have been added, which is 3-day and 6-day pre-incubated chicken feed, meaning that we take chicken feed, we add the water, we leave it for 3 or 6 days to ferment, let's say, just natural fermentation, before we sterilize it with this 2-time autoclave step. And there we could see that for the chicken feed, actually there's no significant difference whether you do this pre-treatment or not. But for the wheat bran, there was something very interesting happening in the sense that we see that after this pre-fermentation, growth restores. We again get normal growth in this model compared to when we have the fresh autoclaved ones. So this for us was a clear indication that this pre-incubation, which is mainly microbial processes happening, rescue our black soldier fly larval growth. At the moment, Nate is very much busy trying to get more into detail. Okay, what could be happening here? What functions could our microbes be doing? And as we do not see the effect on chicken feed and we do see it on wheat bran, we think that microbial biomass on the wheat bran might serve as a source of key nutrients and some preliminary results. But I want to put some caveat here because this for sure needs more research going forward. Shows that indeed that there are two key nutrients, being on one hand protein, which was in this case offered in the sense of casein, and vitamins, which was here the Van der Zandt vitamin mix that we used. If you add both of them together, you see a rescue and even an improvement of growth of your sterile larvae compared to when you have only your vitamins or only your proteins. So it seems to be that the combination of vitamins and proteins is what restores growth. And within these proteins, lysine seems to be even the key amino acid. But again, there's no full reduction here, so this is still research we are now doing. And Nate will also present some research on the Insects to Feed the World conference on this, which is the next step in this research. But of course, still a lot of questions remain in the sense of, does it matter which microbes are present? Can microbes also use too many nutrients? And should we then maybe focus more on optimizing microbial growth on these waste streams rather than larval growth? Or how does this interact with both of them? So for the final 15 minutes, I want to go more on this next step. So I already talked about studying the role of microbes and how we try to tackle that. But you also have, of course, the fate, which is very closely, of course, related. And there we do feel that the pipelines we are having developed for this tracking, that they might help us very much in this front. So this is now what we want to do going further. So there we build actually on this manipulating of the microbiota. So we do have, since I entered, so for the past 8 or 9 years, worked a lot on the fate of microbes. Originally, we were a food microbiology lab, so we looked a lot at food pathogens, and we explored in the beginning a lot of this, what we call horizontal transmission, meaning if you have a contamination in your substrate, does the contamination get into your in your larvae? Yes or no? And is this then possibly a risk for your food chain going forward? Typically, we do these kind of challenge tests where we add our pathogen to our larvae, to our substrate, I must say, in different inoculation levels, and then we evaluate over time, for example, after 48 hours and after 6 days, and see whether our pathogen becomes present in the larvae or in the substrate afterwards. So that's typically the research we have been doing. We have published several articles on that front where we looked at different pathogens. If I have to summarize, what we saw there was that for black soldier fly, we saw a quite rapid clearance of Staphylococcus aureus, a slower clearance of E. coli from chicken feed. And then the interesting part was that for Salmonella, we did not see a similar reduction. Instead, we even saw that it could quite well colonize our black soldier fly larvae. From that research, we, or at least I, built the hypothesis that we have actually 2 factors that you always need to take into account. One being the speed of the pathogen growth in a substrate environment, and the other one being your reduction of your food pathogen in the black soldier fly larval gut. And it's these 2 factors that determine the fate of your food pathogen. Meaning that if you have a microbe that you add that really also establishes well inside your substrate environment, it can multiply in there. Even if there is some reduction happening within your within your black soldier fly larval gut, it will not be eradicated from your your production system, your insect production system, let's say. On the other hand, if you can reduce the growth of your pathogen in your substrate, then combined with the reduction in the black soldier fly larval gut, that might be enough to kick that microbe out of the system. And this is, of course, relevant for food pathogens, but also for microbes you might just want to have. This could be very important. Our key message back then was that you have to be careful with these claims we sometimes saw about black soldier fly being able to reduce food pathogens, because that might be very substrate and bacteria dependent. Now we have been working more on how can we use this to our dynamics to our advantage. And just to show you what one aspect we've been exploring this past year is in the framework of this altering of the fate. So I'm going to show you one example where we are using diet modification or diet component modification, but we could also imagine, of course, that you can use different tools. For example, we have done some tests on phage therapy, which I will not have time to go into today. But of course, you can also just add more of a specific bacteria and hope that this also alters your microbiome community complex. And that you can do to reduce unwanted ones or boost wanted ones. So that's the idea of this alteration. And the example I wanted to show today builds on this observation of salmonella. So here you see the results for our salmonella in the chicken feed. So you see what I said. So we have the 3 days, 0 to 6 days. Of course, in your substrate plus larvae, well, we do not have our pathogens present. It's mainly the substrate plus pathogen. There you do see this increase happening. And in the substrate plus pathogen plus larva, you also see that the substrate has some increase of salmonella, but at least that it's present there, which was not the case if you do not have your salmonella. And then in the larvae, we see a similar increase, meaning that there is a higher count of these salmonella. Interestingly, we repeated this research with what we call swill, which is an artificially made. So we just buy fresh products that mimic supermarket waste. And if we use that mixture and we add our salmonella, we see a different story emerging. So you see here that if you look in both your substrates in the presence or absence of, so in the absence or presence of our larvae, that we have a decrease in our salmonella. And also then, if we look in our larvae, we see that actually at no point we see any uptake of salmonella in our larvae when we add salmonella to this artificial swill. So it's a completely different story than the chicken feed, and we were wondering what could be happening here. And so what we observed is that one of the key differences between the swill and the chicken feed is the pH. So the swill, by design, it's with yogurt, cheese, so there are lactic acid bacteria, so it has a lower pH compared to our chicken feed. So we were wondering if we would mimic this in our chicken feed by adding citric acid, what would be the effect on our salmonella? And already it can show quite clearly, we saw that if we do this acidification, we actually see no counts or hardly any counts. So we do see them, of course, at day 0 when we add our salmonella, but after that, you can see a clear reduction compared to what we saw in our normal chicken feed, meaning that by this type of substrate modification, you can clearly have an impact up the level of salmonella counts, both in your substrate and in your larvae, where we see that we have, it's not a complete eradication, but it's a far lower amount of salmonella that can be retrieved from these larvae. So this is just one example of how by better understanding and mapping the fate of these microbiomes, we can come to tools that we can use to engineer our substrates or our process to reduce these kind of food risks. We're still working on different questions within this horizontal transmission research. So another question we are looking at is spore-forming bacteria, which we know are maybe more likely food pathogen risks. If you think of Bacillus cereus or Clostridium perfringens, they form spores. These spores might impact also how these microbes can persist. So how does that really work is something else we are exploring. At the same time, we want to delve deep. So we have a collaboration with Professor Gianluca Tettamanti from University of Varese in Italy. So both me spent some time in his lab and some of his postdocs came to our lab. And there we are trying to understand the mode of action behind this reduction in the black soldier fly guts. And what we did there, so this is just one of the results we have, is that we went into the different gut sections, trying to understand where this kind of eradication happens. And here you can clearly see, like I said, Staphylococcus is very easy eradicated. And we see that this eradication already happens after 3 hours. And actually we see that your Staphylococcus never makes it through your middle midgut region. So there are some components in your middle midgut that are actually killing off your Staphylococcus and preventing it from passing through your insect, meaning that the factor of eradication is really high for Staphylococcus, which can explain then why we get a reduction in the total system, because the growth of Staphylococcus is not able to cope with this kind of a severe reduction. And a similar story is true for E. coli, where we do see that it can persist. There is some reduction, but not to the same level as what we saw with Staphylococcus. And so trying to understand what actually are these components in the middle midgut is something that mainly Daniele Bruno, who is a postdoc in the lab of Gianluca, is working further on. And then finally, we are also looking at the fate of potentially beneficial microbes. So this is for food pathogens. But of course, you can also say that we have microbes that we want to have because they might improve performance. And on that front, we are working as well. if we look at this research on probiotics, so in literature you can find examples where they show very nice results of adding, for example, specific bacillus species and seeing improved feed conversion or growth performance of our black soldier fly. But if you look at the overall studies that are available, it's very study dependent. So we wrote a review on this in 2023, where we see that often, if we summarize, it's quite black box research, meaning these microbes are added to the system and then afterwards it's evaluated whether you see an effect, but do they actually colonize the gut? How do they grow inside your substrate? These kind of questions were very hard to answer with the current body of literature. So Emma, who is one of my most recent started PhDs, is actually using the setup we have for tracking the plastics in the gut and modifying it to tracking these microbes, as I mentioned, in the gut. And the idea would be there that we try to understand whether we can enrich our microbiome for these fiber degrading microbes. So there are two approaches. You can do it is bottom up, where we use Klebsiella species that we isolated with a high cellulase activity. So Emma has engineered them to produce GFP, so we can better track them. We can then combine them with our axenic model, and we can then start to confirm, okay, if we look after 15 days, do we still retrieve these Klebsiella? Where in the gut do we retrieve them? And trying to answer, okay, do they actually colonize, for example, your hindgut, or what is happening? Do they just pass through? What are the actual dynamics at play here? The other aspect of it is a more top-down approach. So we are collaborating with a group who's actually studying wood-eating beetles. They produce frass, and we can get the frass, which we assume has a microbiome which is more suited for insects that want to degrade high lignocellulose diets. And the idea would be that if we offer this frass early in development, and then we impose a substrate challenge, being a high lignocellulose diet, that we can that the insect itself will select from these frass those microbes that can grow inside the gut and can improve its performance. So this is something Emma just started a month ago with. So also here, caveat that the results are very preliminary. At the moment, it's not very clear whether we can see some effects. This is one graph of one replicate where we do see that at least if we look, so there are two different beetle species, so two different types of frass. And when we add them, we do see a slight increase in growth, but more repetitions are necessary. So there might be some interest to see which microbes are then more present coming from the frass and in our insects to try and understand which kind of microbes might be more suited as probiotics for our black soldier fly. But as a caveat, it's very preliminary. We need more research to confirm this first observation, and then we will study the impact on the microbiota on that front. So as mentioned, I wanted to mainly zoom in on the mapping and the manipulating. Just as a final slide that I wanted to add, it's more for then people who want to watch it afterwards. Our third angle is, of course, all these species of bacteria that we identify might have interesting functions. This can be antimicrobial activity, enzymatic activity, or if you think of the frass, plant growth promoting. So also there we have collaborations between our group and other groups that want to delve into these kind of functions more in detail. So we're always open for collaborations on that front if there is any interest. Going forward, I think I've mentioned most of it. So we really want to push towards that system level understanding of these black soldier fly diets, microbiome interactions. So as I mentioned, our insects are a bit different compared to traditional animals. It's not just a feed going in, it's a feed passing through, being reintroduced. So there are far more interactions. So we need to look at the microbiome, not only in the guts, but also in the diet. And how it changes over time is important there as well. Quite recently, we're also more intrigued in this genetic engineering, not only of our bacteria, but also of our black soldier fly. And we're trying to set up a collaboration with Professor or Dr. Paul Mozdiak, who has experience with this, because we think it might also be a way to get better tools to use our black soldier fly, for example, as a bioincubator, to process different diets, but also from fundamental point of view, if we can knock out certain immunity genes, how does that impact our insect microbe interactions is also something we want to explore in the next, let's say, mid long term agenda. And then, of course, just keep on pushing to really assign functions to members of the black soldier fly microbiome. I do hope that in 10 years I can definitely say what Providencia and Morganella are doing. And I think we're getting closer and closer to doing just that. With that, I like to thank the colleagues over the years. So a lot of people contribute to all of the studies you have seen. Also, a lot of collaborators from around the world collaborated, helped us to achieve this. And of course, a number of funding agencies as well. And I want to thank you for your attention, and I'm happy to take any questions. And if you want to know more, you can follow us on LinkedIn, where we push for new research updates as often as we can.
Wonderful. Thank you so much. That was a lot of fantastic information. I see a lot of hand clapping in the emojis, and
Thanks.
I'm sure that we will have some questions. So you can put your questions in the chat or raise your hand. And Jeff, you are first up.
It was an excellent presentation. I have a lot of questions, but I'm just going to start with this one is with the azenic larvae. Do you think there's a microbe that's part of its core microbiome that's really essential for like establishing the morphology of the gut to allow it to operate? Because I think of Gianluca's work in exploring the gut phenotype.
Yeah.
So if you go with an azenic, I assume the gut's going to be highly reduced in terms of its morphology.
So actually, I think I can say definitely that that's not the case, in the sense that we collaborated with Gianluca as well on that research. So Nate went to his lab and we've compared the functional, morphofunctional of both our Axini gut and the let's say wild type gut. So we have done the same type of analysis we've done in the past, and we don't really see any impact on the gut morphology of the presence or absence of these microbes. So there were no clear differences that they found, telling or worrying on that front. We could see that there were no microbes present, so it was clear in that sections, but there were no really apparent differences between the two.
That's pretty interesting because hasn't it been shown like in vertebrate models that when you do azenic work like on mice, the gut morphology does change?
Yeah, I think like microvilli depth and so on could be
Yeah.
influenced, but there was nothing really apparent in that. But yeah, we're wrapping up that paper also at the moment. Nate has to push it out, but there were no apparent differences, at least in what we did. Of course, it's just on these are bred on the chicken feed, where we also do not have a really big impact. Maybe it's different on different diets. I think there's for sure the diet component in there as well, which of
Okay.
course complicates things, but that's always the case.
Very cool. I want follow up and then I'll stop. I do have a list here, but I'll stop after a second. With the amino acid work that was done, the work that was published a couple years ago, that's based on the insect response. But what about the microbiome response? So you feed these amino acids different concentrations, what's the microbiome doing? Has anybody taken a look at that yet?
No, and to my frustration, it wasn't, and that's actually what Cato is the so yeah, that's the fortune of starting as a PhD as a professor. You get some extra push. So I have another PhD student who's actually trying to answer that question, because if you look now, you often have like characterization, and we are guilty as well because we did in the past as well. We look at the microbiome in your substrate, and you look in your microbiome in your FRAS, but you don't look at what happens at day 1, 2, 3, 4 in between. So how it changes over time, and how indeed the microbiome reacts to decreases or increases in the amino acid levels and so on. I don't find, maybe I'm not up to date, but I haven't found good studies on really that in-depth understanding of like how does it actually change, or how can we link it to amino acid levels and so on. But for sure, I think that's, yeah, if you look at really fundamentally understanding your system, it's something we need, but it's a lot of work. Yeah.
Well, I appreciate what you're doing to promote the black soldier fly as a model organism for others to
I,
explore.
I
So
keep
this
I
is
keep
great.
pushing that indeed as much as possible.
Cheryl, did you stop recording?
No, we have more questions.
Okay.
So Eric is next.
Thank you so
You
much
ready?
for doing all this today. It was absolutely fascinating. I'm sad that I came late because I'm sure I lost a lot of information. So I'm looking forward to that recording. But I'm thinking about the Asiatic model, because whenever I was doing my master's degree almost 10 years ago now, I think they were doing Asiatic models in Aedes aegypti or some other vector-borne disease work. I don't know if I captured everything, and I don't know if there's a real question in here, so I apologize. But I know whenever they were trying to do fecundity with Asian models, they were having issues with fecundity or second generations of mosquito work. So I didn't know if you were just trying to have that Asian model to do a single generation, but I didn't know if the multi-generational studies or the evolution of it
Mhm.
as well, how if there's plans for that or how much is that being affected? Because it saw that you had grown them up for a certain amount of days, but I'm curious about second or third generations.
So at the moment, it's a single generation exenic model. So we use the larvae and then we halt it. So we did check whether we can gut to flies, but then yeah, the tricky thing is set up like how do you keep them sterile as flies? They're a bit bigger as fruit flies, so your container would need to be bigger and still sterile. So there are some practical hurdles that we are thinking about because we do at least want to show it. Would be nice if we could keep it indeed sterile for longer terms and not have to treat our eggs because we're not sure what the impact of that treatment might be as well. Um, but because we're mainly, yeah, you have to choose at some point, and we're mainly interested in this microbe interactions. I'm not sure how far we will go in like multiple generations and fecundity. I do think we really want to go to show that that's not impacted, so that maybe other people can work on that further, but it's not a main focus of what we are doing.
No, that's no understood. I also have more questions, but I will digress a little bit. I will comment that I'm glad to see that there are individuals working out there with different types of waste streams or wanting to look at that and see how the microbiology looks like over time. Because I also had the idea many years ago of you're going to raise specific lines of black soldier fly for specific types of waste streams. And over time, that would be one way to go. So it's very fascinating. And then lastly, the automatic monitoring, looking at the heat maps of the actual bins themselves. I was always interested in seeing how, like those hot spots over time, how that's affecting their microbiome. Because if you're raising them at different temperatures, you're going to have different internal temperatures of the bin. And that in and of itself is going to have impact on pH, impact on microbiota, not
Yeah.
only within substrate, but within insect as well. So it's
No,
fascinating seeing that.
yeah, I fully agree, and that's also in the idea of Cato, her PhD. So we're now doing, so it's a combination with the PhD that is also looking at automated monitoring. That we have these large crates, and she is sampling both active, meaning hot locations, and inactive, meaning cooler temperature locations, and seeing how whether we can see differences in both nutritional composition and microbial composition. So trying to see if what is actually going on and how heterogeneous the entire mixture is something we try to. I think, yeah, whether it will be practically relevant, that might be the next step, but for at least for fundamentals, we want to understand what is happening there.
I look forward to seeing it coming out.
Will take some time, but I hope to, yes.
Wonderful. Any more questions?
So I have a follow-up. You referenced the Douglas paper from 2020, the pie crust paper. Have you had a chance to use it?
Pie crust, I have to double check. Actually, I have to discuss with Freek. I think we're a bit shy on it. I think he run one of an analysis, but yeah, you can see a lot in that and you can get a bit lost. No, we had our same issue because we also did like a whole shotgun genomics analysis, so you get a lot of data in there as well. But
Right.
what is what? It's so it's not like we have it routinely running PyCrust, but
That's I think I get you. I introduced you to the group from Berkeley that's doing some of the shotgun sequencing.
yeah,
So there might be a chance.
those I've seen at BSFCon, I think as well, or at least I gave a talk there as well. So I had some, yeah, but also interesting because I think they also are running this meta-analysis, which I haven't seen out yet. So I'm curious to see where they are with that as well.
Really cool work. I mean, really, really excellent research.
Thanks. We try.
No, well, it definitely shows. You're doing a great job. I mean, I think do you think there's opportunities to lead this towards human health? Like Black Soldier as a model to explore different things?
Um, well, I'm not sure. It does have the benefit of, I think, like you can really work on more relevant diets compared to what's been done in Drosophila. So to
Mhm.
some extent, I would see that, but if need be, then I would have to collaborate with people who have more of a human touch on that. So at the moment, we're not really claiming that too hard, but the fact that you can work on diets which are more like human diets, I would assume would be very interesting to have a
Yeah.
simple model to better study gut, diet, microbe interactions in that front. But yeah, I
We're
haven't
not
had
there
much
yet,
discussion
right?
with colleagues yet on that front. No,
I think I agree. We're not quite there yet, but once we get to the build up momentum and we can engage, I think that'll create opportunities for funding beyond what we traditionally get.
let's hope so indeed.
Yeah, hopefully.
We have another question from Michael.
Yeah, Jeroen, thank you. And sort of echoing everyone's enthusiasm for the presentation. It's great. A lot of insights in there. I just figured I'd toss it out there. You showed some of the other research areas. Anything you want to cherry pick from the enzyme work to give us a teaser on anything coming down the pike? Very curious about that whole avenue.
No, well, we have some. I think we are the first to along, and now especially with Emma still working on it, is on these Klebsiellas with like hemicellulase, cellulase activity, trying to understand. Well, the next step would be that we sequence their genomes to see what kind of cellulases, hemicellulases are there. Are they really different? Might there be something innovative there? But also there, well, KU Leuven, thank God, is a large university with a lot of people I know. So there are groups that really focus on then in vitro expression and so on. So this will probably not be something that we can do ourselves. But the first step is really trying to identify these. So it's not something that's ongoing at the moment. But I think if we do something on these enzymes, it will be there. We have done some more antimicrobials. But then that's also with the University of Antwerp and also with AMPs. But then you get into peptides as antimicrobials, which is not ideal. Then with molecules would be better. So at the moment, there's nothing really concrete. But if it would be something, I think it will be cellulase, hemicellulase.
Well, that brings me back to the lignocellulase from the beetle frass research you shared, and kind of tying back to Eric's question in a different context. If you see changes in a single experiment there, are you going to go to multi-generational if you see the kind of ability there around lignocellulose and see the effects over time?
Yeah. To be fair, at the moment, because we have done in the past, there was a different PhD student who already tried, and it's not that easy to get PSF to really behave well on like high lignocellulose content. So we haven't seen something promising yet that would warrant us like here we have to go much more in depth. So I would have to see something more promising before I would say yes, let's dive into it. And if it's my hope is if it's microbial, then you could not sure if it would then enhance over different generations or it's something you can have as a treatment and then might as well be that you add microbes that do a lot. I'm more and more believing in you can improve within your insect, but you can also improve within your insect rearing. You know what I mean? In the bioconversion
Sure.
process as such. So maybe we just need to improve the bioconversion process and then the BSF can benefit from that rather than we have to improve the BSF. And then maybe by understanding which microbes could work in that kind of system might be an easier, more straightforward way to improve the process than really having a BSF that's good at digesting lignocellulose because it's really at the edge, I think, of what this bug was supposed to do, or that's at least my idea.
Well, we are just about at time. So with that, I will pause the recording.